everybody. My name's Meredith Gore. I'm a professor over in geographical sciences and was a faculty affiliate in your college for a number of years. I had the good fortune to collaborate with a number of faculty and also graduate students and undergraduate students in the department. And so I am very fortunate to be able to join you all today. I really appreciate your time and you all helping me welcome Dr. Diego Cardeñosa from Florida International University. Diego and I have the good fortune to collaborate with Diego on a really cool National Science Foundation grant, and a lot of the research associated with that grant is going to be discussed today. But a little bit of background about Dr. Cardeñosa. He is a scientist using molecular and forensic tools to fight the illegal trade in endangered sharks from Hong Kong to Latin America. He's done a lot of work to understand small shark fins, many of which are taken from protected species. And he's also done a range of collaborative science with multiple governments in Latin America to help develop new protocols for interrupting the illicit trade in sharks and other species. I'm going to ask Diego to say, well, thank you so much for coming here today. Thank you for sharing your research with us. And we're really excited to have you here. Thank you.
Thank you, and thank you guys for coming. So yeah, like Meredith was saying, I've been working in related topics, especially towards the international shark fin trade and everything that surrounds it, from a global perspective to a very narrow location like is Hong Kong, where everything is funneled in, at least in the trade for shark fins. So let's start with, see if which one would work.
There you go.
Yeah, just let me hide these things again. So a little bit of background: conservation biology is usually considered a crisis discipline. And as a crisis discipline, we need to understand the processes and how it works, what makes it such a crisis discipline. That part manifests itself in the number of species that are threatened by extinction. Here we have just a couple, but there are over 100,000 species between animals and plants that are in any of the threatened categories by the IUCN. That would be vulnerable, endangered, or critically endangered. So we need to understand that a little bit more and not just get a little bit overwhelmed by the global crisis that this is. We need to make quick decisions as scientists with the current available data. And that's usually where the heightened sense of crisis comes, because a lot of the times to make those decisions, there's really no available data to make them. So we need to develop them and we need to create them. And very little taxonomic groups envision that crisis as sharks. So this is an oceanic white-tip shark, part of what we consider pelagic elasmobranchs. In the last decades, since the 1970s, over 70% of the populations have declined. So there's a 70% decline in the populations of pelagic elasmobranchs. And the reasons behind that is an 18-fold increase in fishing pressure since 1970. So imagine the high seas. It's a very, very large expanse of ocean, and that's where we have been able to affect this group of sharks to the extent of 70% in declines. But that's not only that. Even the coastal sharks, or I think the coastal sharks are even worse than the pelagic sharks. Over 1/3 of sharks and rays and chimeras are threatened with extinction. But if you look at the tropical and subtropical areas, 3/4 of the species in those locations are in threatened categories. When that paper came out in 2021, those 3 species were considered close to extinction, but now this ray, the Java ray, has been declared extinct, making it the first marine vertebrate ever to be considered extinct due to anthropogenic reasons. So what's left, you can take a little bit of panic and anxiety directions, but I think when I started on my biology career, I thought of science as, let's say, my weapon of choice to combat some of these crises. To me, science provides us with all the tools that we need to revert some of these global trends that we're seeing, but we need to understand really well what's going on. So the first thing we need to understand, what are the proximate threats and what are the ultimate threats? By proximate threats, I mean what is directly impacting the populations of sharks and rays. For, in this case, overexploitation or fisheries. Overfishing is the number one threat of for this group of species anywhere in the world. But that threat, or the overfishing, is driven by a global demand as food and a global demand of shark fins from Southeast Asia. So these two, we need to find the connections between these two threats. What gaps do we have in knowledge in order to connect them? And how can we get from threats to an actual conservation and sustainability goal? So the theory of change for this between proximate threats and ultimate threats, the connection is to understand the supply chain. We know that there's a global demand, there's a global supply of shark fins, and we know it supplies the large shark fin markets in Southeast Asia and specifically Hong Kong. What we don't know is which species are being traded. Are all species trading the same volumes, or there are some species that are overwhelmingly more common in the market than others? Where are they coming from? And is it legal? The part of where they're coming from seems trivial, but it's not, because when you see the statistics by the FAO, a lot of the trade on ILASO ranks is shark and ray, but you don't know 100 tons of shark and ray, is it the critically endangered scalloped hammerhead, or is the near threatened blue shark? Or if you don't separate them by species, there's not really much more than we can do with those statistics. So let's understand a little bit more the complexity of this. This is a very, very simplified map of the sources and the locations where shark fins are taken from and going to, but most of the arrows point at a single location, which is Hong Kong. But when you see the non-shark fin commodities, which in this case would be mostly meat, you'll see that South America, and specifically Brazil, is the #1 consumer nation of shark meat. So whatever China means for shark fins, Brazil means, it's the same for the shark meat. It's around 80% of the traded shark meat is consumed by Brazil, by the Brazilian population. So most of you might have seen this type of picture. Most of us will see it and think about shark finning, which is the practice of catching a shark in the ocean on your fishing boat, cut the fins off and throw the body overboard. The reason behind it is not really because fishermen are evil people. Most of the reason behind it is, imagine this room. If we wanted to catch sharks and we bring them to our freezer, we could only hold, let's say, 100, 200 sharks in this room, frozen. If we cut the fins, which are the most expensive and most valuable part of the shark, we can capture tens of thousands of sharks and just fill the room with fins. So the shark finning issue is just created by the fishermen to trying to maximize their profit in the same fishing trip, just by filling their freezers with fins and not with meat that is much less valuable. But here you have a picture that is not that too long ago. This is basically what's going on in large fishing ports all over the world, and you see all those sharks have their fins attached. So not only by solving the shark finning issue, you're going to solve the overfishing issue. And then you see a lot of campaigns online saying stop shark finning, but I'm telling you, if we stop 100% shark finning right now, we wouldn't be solving the conservation crisis. We will be solving an animal welfare crisis, but the conservation, even if we bring the sharks on board and to land with the fins attached, it's still pretty much overfishing of their populations. Once you land the shark, you can cut the fins, you can put them on a rooftop. I took this picture in a drying facility in Ecuador, in Manta, Ecuador. Most of these fins are pelagic thresher sharks, an endangered species, and the floor was just covered by them. So they leave them outside at the sun for 2 weeks to keep them completely dry. And that way you can trade them without having to freeze them. And it's an easier way to not spoil them and be able to store them for longer periods of time. Once they arrive to China and Hong Kong, they remove the skin, they remove all the muscle and everything else that might be there, and they just leave what we call the ceratotrichia, which is the cartilaginous tissue that holds the fin, basically makes the shape of the fin. Then you can go to the markets and see fins like yellow fins all over the market. But just by looking at that, you don't know which species is this. You know it's a shark fin, but is it a blue shark? Is it a silky shark? Is it a hammerhead? You have no clue just by looking at it. Then those fins end up in shark fin soup, which is a very highly desired dish in Southeast Asia. It was considered the food of the emperors back in the day. You would be considered very wealthy if you consume shark fin soup. But with the rise in the economy of China, more people can have access to shark fin soup. The demand increase, but the supply of sharks basically remains the same, right? So that's what's driving those population in decline, like I showed you before. So just to give you an idea, I took these pictures in my last trip to Hong Kong a couple of years ago. And in these jars, there was one that caught my eye that is for one catty is around 600 grams. It costs 7,800 Hong Kong dollars. If you put that into US dollars, is 1,000 US dollars for 600 grams of shark fin. So you can see why people are drawn towards this trade because it's very, very profitable. Also, another thing that you will notice when you go to the markets is that all fins have the same shape, the same cut. That's not because all shark fins have the same shape, the same length, the same, it's because when they're processed, the traders trim them and they generate these what we call shark fin offcuts or trimmings to make it look better, more visually appealing, and remove parts that are undesirable for the high-end soup. But these ones are sold for a lot less than the fin. If we wanted to do a survey based on large shark fins, there's not enough NSF grants that we can get in order to go to Hong Kong and do these surveys. So we use this as a proxy towards being sold in the shelves. Through all that process, from catching the shark to selling it in the market, the fin goes drying in the sun, it goes chemical processing, so they use bleach and other things to make the final product as pretty as I showed you in that picture, like a very unique shark fin. And the DNA degrades through that entire process. So if you want to take a sample from one of those fins and amplify the entire cytochrome oxidase 1, you wouldn't be able to do it. You will succeed maybe 1 in 100 samples that you run. So we designed a mini barcode assay that amplifies highly polymorphic and highly informative regions in the cytochrome oxidase 1, 150 base pairs or 200 base pairs. And when you combine them, you can get basically every single elasmo rank identified with a combination of those two sequences, and we can apply it to shark fin soup, liver oil pills, several products for skincare, because the liver oil of the sharks are used for skincare products as a moisturizing agent, as I understand. So it's a very powerful tool, because we can bring some of those products to the species level and identify what's being used there. And that led to basically an explosion of scientific work that we've been able to do based on those Hong Kong market surveys that we do. So which species are traded, and are they traded the same? This is more or less what we've been able to accomplish so far. It's the largest data set on an international shark trade data set that you have. We have 1 decade of an interrupted survey. This started in February 2024 and is still ongoing. I think the survey for September this year is being conducted as we speak. We have identified I think this is up to 2022, but I received the data for 2023 and 2024. So I think we passed the 20,000 samples identified to the species level since 2014, and we have identified 106 species that are present in the markets. Before, we only knew from Shelley Clark's papers back in 2006, around 14 species that she was able to identify in the fin trade. But now we are to 106, and we have a much better idea of what's being traded in which quantities. So here you have two plots. I'm gonna explain this a little later, but you can see basically which species are the most common in trade. Here is very interesting that only 2 of the top 10 species are not in threatened categories. Number 3 is a critically endangered shark. And this shark, this is for offcuts, and this is for a different survey that we've been able to conduct on small fins. You can see that the scarlet hammerhead is among the most common, and it's a critically endangered shark. And we're basically trading on juveniles and on adults, so we're not leaving a lot of room for that population to bounce back, basically. So now that we know what's being traded in what proportions, we can try to identify where are they coming from. So we've been able to publish some papers about this, and we basically name it DNA zip coding, because we can take the samples that we have identified to the species level from the markets, and through mixed stock analysis and population genetics, we can tell where they come from. So just to give you an idea, here we have the distribution map for the pelagic thresher shark, and there are 3 RFMOs, or regional fisheries management organizations, that see the high seas fisheries in all that location. So you have the Indian Ocean Tuna Commission, the Western Central Pacific Fisheries Commission, and the Inter-American Tropical Tuna Commission. For the silky shark, it's different. It's a much more globally distributed species. It occurs also in the Atlantic, where the International Commission for the Conservation of Atlantic Tuna sees it's kind of like the purview of that geographic location. And we have samples from all these places around the world, so we can have an idea of the haplotypes that occur around the world, their haplotype frequencies in these different locations, and we can use that information to trace some of the fins of Hong Kong to a region of origin. For the pelagic thresher shark, in a paper that we did back in 2020 with data from 2014 to 2017, we can see that 85% of the pelagic thresher shark fins that we saw in the market came from the Eastern Pacific, and a lower proportion in the Western Pacific. Now we're working on a different paper that takes into account a full decade of data. And here you can also see the volumes that are being reported to CITES, which is the Convention on International Trade in Endangered Species. And we can see that the Western Pacific, only one nation, Indonesia, in 2021, reported trade of pelagic thresher shark in 2021. So basically what we can see from here, here, and here is likely illegally traded pelagic threshers that arrive to Hong Kong without the required CITES permits. So all sharks that are in CITES, is 149 species, are in Appendix 2. That means you can still trade with them, but in order to do so, you have to have permits that basically show that they're legally caught, they're traceable through the supply chain, and that the volumes that you're trading is not detrimental to the survival of the species. If you can corroborate those 3 things, you can still trade with that species, but it's impossible to have this amount of thresher sharks in the markets of Hong Kong with only one country reporting to CITES. So it's kind of a clear evidence of continuing and ongoing illegal trade for sharks. For silky sharks was a little different. The population genetics based on the mitochondrial control region tells us there are 2 populations, one in the Atlantic, one in the Indo-Pacific. All of the samples that we found in Hong Kong fell within the Indo-Pacific population, but some of these haplotypes are shared between the 2 populations, meaning some silky sharks that have been caught in the Atlantic have that haplotype. So we we needed to do an extra analysis in order to know or estimate where they came from. We use mixed stock analysis, which compares the mixed population, which will be the market, and the haplotype frequencies from the market, the haplotype frequencies from the known potential source populations. And based on that, we can have an estimate of where they come from. At least for silky sharks, all of them came from the west in the Indo-Pacific population and none from the Atlantic. Why is that? Since 2013, ICAT, the RFMO from the Atlantic, has a no retention, no landing, no trade on silky sharks. So what we think is a rare case of high compliance by industrial and high seas fisheries with this, but we see for pelagic threshers and for scalloped hammerheads that is not the case. So at least silky sharks in the Atlantic are well managed, and there's high compliance with regional laws that are put in place for that. So how about the legal framework? As I explained before, there's CITES. For those that are not familiar with it, it's the Convention on International Trade in Endangered Species. It has two different main appendices. Appendix 1 bans all commercial trade for species that are in Appendix 1, and Appendix 2 is what I told you. So if you can have the permits for legal acquisition, non-detriment findings, or meaning that the specimens that you're trading are not detrimental to the survival of the species, then you can do so. The first sharks that entered on CITES were the great white, the basking shark, and the whale shark. Flagship species, very charismatic, big, but not really common in trade. In over 20,000 samples that we have analyzed in Hong Kong, we've never found any of these ones. So it was good that they enter on CITES, but they're very heavily protected everywhere where they occur. And it was a big hurdle to try to break the barrier to list commercially important species into CITES. One of those reasons was because the usual suspects, China, Japan, and a lot of the Southeast Asian nations said that if they receive fins from a shark, they couldn't tell if it's one species or another one. So they couldn't have good enforcement and compliance if they enter CITES. But a group of scientists came together and made visual identification guidelines for fins of oceanic whitetips, port beagles, and hammerhead. If you see an oceanic whitetip shark fin, it's very obvious it's an oceanic whitetip. Rounded apex with a white marking at the tip of the fin. So based on this, they were able to show at the conference of the parties that you can actually identify fins from these species very, very simply and fast. And for the first time in 2013, these 5 commercially important species were listed in Appendix 2. So if you think about our Hong Kong surveys since 2014, when those species were starting to be enforced, we have 3 of the most common species in the markets on CITES. Great. 3 years more, in 2016, the pelagic thresher, big-eye thresher, common thresher, and silky shark were proposed to be listed in Appendix 2 based on mostly on the data that we were collecting from the markets. They can look at this list and say, okay, what other species can benefit from being listed on CITES? So now we have the silky and the pelagic thresher there listed since 2017. Then 2019, Makos made it to the CITES Cup. Parties, again, they voted in favor of it. So one more species in the list of the most common on CITES. But then, what then? We were kind of getting into those large pelagic sharks listed on CITES. We published this paper where we saw that between 2014 and 2021, we saw an increased number of endangered, critically endangered, and vulnerable species making it to the markets. Almost all but one of the top 10 species were in that threatened categories. And if you see the species that fetch the highest prices in the market, most of them were critically endangered. So that kind of gives you an idea of this ongoing issue related to the trade. So here we have this plot. In the y-axis, we have incidence, or just basically how common you are in trade. The higher you are in the y-axis means you're more common If you're really low like these guys, you're not that common in trade. And here we have FAO major fishing areas. So the entire world map is basically divided into 16 major fishing areas. So the more to the right you are in this plot, the more cosmopolitan you are, and the more to the left, the more restricted in geographical range you are. So we can see that what we have in CITES at that time, before 2022, most of the CITES listed species that we had were here. So cosmopolitan, common in trade, and with a mix between vulnerable, endangered, critically endangered species. But what's happening in this quadrant? So here we have critically endangered species, endangered, vulnerable, very geographically restricted. So this one only occurs in one single location around the world. It's over 1% of what we see in Hong Kong. And you say, oh yeah, but like, what's 3% of the market? It's nothing. But when you think that 6 million kilograms or 6,000 tons of shark fins arrive to Hong Kong on a yearly basis, a 2, 3, or 4% of that is a lot, especially for a critically endangered species, and especially if that critically endangered species only occurs in one single location in the world. So it seems not too much, but for these species is a lot actually. So on that paper, we tried to make the case that for the next CITES species that were going to be proposed, we should look more into this quadrant of that plot. So that led to the COP19 in Panama in 2022, which was kind of historic because one entire family was listed under Appendix 2, and we passed from having 50 species of Elasmobranchius to 149. So the number of species there increased tremendously. So now we have black tips, the male shark, blue, basically every single one of those common species that we see in the offcuts, in the offcut surveys are now on CITES Appendix 2. But in 2018, I moved to Hong Kong to live there for a full year. I was going like 2 or 3 times a year during my PhD, but I spent all my 2018 there working with the authorities and the custom authorities and the environmental authorities of Hong Kong to see how can we enforce all these problems a little better. One of the containers was unpacked, and what I saw was a lot of these sacks just put aside. They were put under X-ray and then just put aside. I was like, why are you putting this aside and you're not looking at this? These are full of very, very small shark fins, smaller than the palm of your hand. So there's no way to visually identify those fins to a species. So there's nothing they could do. They just didn't look at them at all. So I went to the markets dressed as a tourist with my I Love Hong Kong hat and t-shirt, sunglasses, camera, and I went through the markets, the street markets of seafood in Hong Kong, and I started looking for small things. I was not looking at the big ones. I just wanted to see if I could find the little ones. And there were jars and jars and jars full of processed small fins. So that was a little bit of an alert. Here you can see, I opened one of those, that's my hand, and there can be hundreds of thousands of fins in one single sack. And nobody has ever looked at this before. So it was a big problem. Last year, we published the results of the full survey that we did in 2019. And we can see that a lot of the species that we see in that survey are also in threatened categories, but the composition is very different, right? Here, blue shark, you don't see blue shark. I don't think you might see silky, but instead of being number 2 is right down here. So a lot of the species that are here are small coastal species that don't grow more than 1.5 meters, but you have some similarities. Scalloped hammerhead, the silky, as I said, I think, what else is big there? Spinner maybe, and smooth hammerhead. So the fact that those sharks are in this survey is because they're caught when they're juveniles. So one of the things that caught my eye was like, I think this size, and when we analyzed it, it was a scalloped hammerhead. When they're born, the fins are this big. So it's potentially that they caught a pregnant female, they caught the fins of the little babies inside, and and made it to the market. So it's a big problem especially for those threatened species. We did basically the same plot, and we saw that what we thought it was not that common in the offcuts is actually a lot more common in the small fins. And the reason behind it is because small fins don't get trimmed. They don't generate offcuts of small fins, otherwise they cut the fin in half. So the representation of these small species was being obscured by the survey that we were doing on the offcuts. Now, by doing offcuts and small fins, we have the real representation of the entire fin trade. So here are those species. Most of them are from a single family called Triachidae, or the Triachids. And those are being proposed to be listed next November on Appendix 2 in the next CITES Cup. And like this paper was feeding directly into that proposal, saying, hey, we haven't looked at these categories before. Look what is being here. There's critically endangered species in that group of sharks, and we need to do something about it. And all these are also part of the small fins, but as I said before, most of these are just juveniles that we are bringing to the market as well. So all of the black bars that you see in these plots are species that are on CITES. The blue ones are not on CITES, but these ones, if they pass, they will make it into Appendix 2. And if we are able to push that proposal forward, it's like putting a lock on the shark fin trade, meaning every single shipment, consignment, container, anything that is coming through a border that is sharks, they have to come with a CITES permit. They have to come with a CITES permit. There's no way that you bring a container from Latin America to Hong Kong without CITES permits, because basically everything that is fished in those waters is on CITES. So going back to our theory of change, understanding the supply chain, we now know what species are being traded, in what volumes, and for some of them, where they're coming from. We have been able to increase international trade regulations. So back in 2013, as I said before, there were only 5 species in Appendix 2. Since 2022, we have 149, and it basically follows all the evidence and all the data that we've been able to generate with the survey in Hong Kong. But now, in order to go back to that conservation sustainability goal, we need to ask ourselves, is the ongoing trade legal? Is their compliance and enforcement adequate? So we took back again the data from the Hong Kong market for those first 5 species that enter on CITES, and we compared what is being reported by the CITES parties, and we compared the 2014 data set, which was pre-enforcement, and the 2015, 16, and 17 data set that we have by then, to see if there's any change in the proportion of those CITES listed species in trade. And we see that they were an order of magnitude higher than what we would expect based on what is being reported. Here you can see there were 20 tons, a little over 20 tons reported by CITES parties to Hong Kong, but that only corresponds to 0.4% of the total imports of shark fins into Hong Kong in that year. So if you go to the markets looking for those sharks, they would be very, very rare in a perfect world. What we found out is that they were very common. They were around 10 or 12% of our surveys. So again, a clear evidence that enforcement and compliance was quite low. So how about now? In this bar plot, you can see the amounts and the percentages that correspond to the total imports of Hong Kong since 2015 to 2021. And you can see that the total imported volume by Hong Kong is dipping. So you can say, oh, awesome. We have species now on CITES. It's going down. So that means that decline has to be because CITES listed species are not making it to Hong Kong and everything is working well. But when you see how common those species are in trade, at least for the first 5 that were listed in 2013, we only saw a significant decline in poor beagle sharks. So they basically disappeared from our surveys since 2017. The reason behind that, we believe, is because that species lives in the US, in Canada, in New Zealand, in Australia. Those are high capacity nations that you would expect that they have high compliance, high fisheries management. And actually, they're probably the only 4 countries that have well managed shark fisheries. So that was to be expected. The problem is that these other 4, we don't see any significant change since we started the survey. Those are tropical species, cosmopolitan species, where no fisheries management or compliance basically exists for them. We did also a little bit of that DNA seed coding for the scalloped hammerhead shark, and we can see that the only regions or populations or countries within populations of the scalloped hammerhead that were reported any kind of volume or trade to CITES were the Eastern Pacific, Western Pacific, and Arabic Peninsula. But here, the Central Pacific, Southwestern Indian Ocean, Eastern Atlantic, Southwest Atlantic, they have contributions to the market and 0 reporting to CITES. That means clear evidence of illegal trade. But also, if you see here, the Western Pacific, 2 nations within the Western Pacific were reporting to CITES, and it was only 1,000 kilograms, 1 ton of shark fins. That 1 ton cannot represent these high volumes that we're seeing in the markets for the scalloped hammerhead. So there's obviously a mismatch, a massive mismatch of what we're seeing in the markets and what we're seeing being reported by the different nations to the convention. So how can we tackle this problem? Now it's a matter of, we have the evidence from the markets, how can we make this better? What tools are out there that would allow us to improve the situation? So back in 2018, we did this. Let's see if, yeah. So what that does is uses species-specific primers for different species, specific forward primers for those of you familiar with genetics, and a universal reverse. So when you combine all those primers into one single reaction, you get different size fragments if the target DNA is there. So basically, when you run that test, it gives you, if you have an amplification curve, means that inside your tube, you do have DNA from your target species. If you have a flat line, that means it's not one of those species that you're targeting. But it also, based on MEL curves, you can identify, for those that amplify, which one is it. So I worked with the Hong Kong authorities in Hong Kong in 2018. As I told you, I moved there. First container, small fins, they were gonna release it. I told them, just give me 3 hours just to, to prove that this works. I tested 94 different samples from different sacks, and I went as fast as I could, extracted DNA in the field, ran the DNA test in the field, and 3 species lighted up, and they end up being a scalloped hammerhead shark. So I told them, listen, you were not looking at this container. It does have CITES listed species in it. Then another container came with sea cucumbers and processed shark fins. Processed shark fins meaning the yellow fin, no way to know what is there. Run the test, again, something lighted up. when we saw scalloped hammerhead. Then a large container from the UAE, United Arab Emirates, 16 tons of shark fins worth 400,000 US dollars. We went through it, silkies, threshers, hammerheads, the whole thing was there. And that basically led to a big seizure by the Hong Kong authorities. Once I proved them that this could work, they were like, oh, we have problems with the European eel. There's a lot of smuggling happening through our borders. It's basically main use for sushi, unagi. And this is basically fishermen all over the place in Europe and in the United States, in Maine and other fisheries, trying to capture glass eels when they're coming from the ocean to the river. Then they sort them, put them in a bag, and send them to China, where they grow them to be an adult and then for meat consumption. So they were very interested in the European eel. I developed a species-specific primer for the European eel, and then one Saturday afternoon, I was at home in Hong Kong watching Netflix or something, and they called me, we have 2 smugglers, 4 suitcases, come to the airport now, because we need to figure this out. So I took my little kit, my DNA test kit, went to the airport. When I went there, I found this out. You can see here little worms swimming in those bags. So it was 4 suitcases full of that. And when we went there, took some of those glass eels, even water from the bags that they were swimming in, and everything lighted up as European eel. And that led to the first ever prosecution of an eel smuggler in Hong Kong history. So they were like, oh my God, this really works. And they've been using it since, and they see a decline in cases of smuggling of European eel. We've tried this in the US last November in New Jersey, and we found containers coming from China to the US labeled as European eel, and it had labeled as American eel, and it had European eel in them. So very useful tools for law enforcement. Same for this turtle, very charismatic, the Mata Mata turtle. It lives in the Amazon and the Orinoco River Basin, but here in Leticia, it's a big transshipment point to smuggle them into Brazil and Peru. When they capture them, they don't know where they came from, so they want to release them to either location, but if you release them in the wrong location, you might be playing around with outbreeding depression of the populations that you're introducing. So we created a test that tells you whether they're from the Orinoco Basin, the Amazon Basin, and within a couple of hours, you can know where they come from and try to do a reintroduction quick, instead of having facilities with thousands and thousands and thousands of animals that just have a painful death there, or they never actually get released to the region of origin. So I'm gonna move forward quick because I'm running out of time, but are species-specific primers the way forward? Since 2022, I told you we have a lot of the most common species in trade, at least for sharks, on CITES. So I don't desire anybody to do 149 species-specific primers, because that would take them a whole career, a nightmare, and a bunch of sleepless nights. So there's another way. And then we need to start looking at the trade in small fins as well. So we developed something called DNA, which is part of the project that I have with Meredith and other colleagues. And we moved into something called high resolution mel curves. So basically it's the same qPCR, but it detects little nucleotide changes in an amplified sequence of DNA. So what we looked for is a highly polymorphic location where there's a lot of different nucleotide mismatches from different species, flanked by conserved regions or locations where we can design universal primers. So this tool uses a universal reverse, a universal forward, with a high polymorphic location in between, and the result is different curves, mel curves, and high resolution mel curve for each individual species. And it's consistent. Every time you run a silky shark, every time you run a blue shark, it gives you the exact same curve. So this curve is a visual representation of the nucleotide composition and organization of those nucleotides in that particular fragment of DNA. But we didn't want you to go through, okay, I have this curve, let me go through this booklet and see what it matches. It will be not efficient. So we partner with our colleagues at Worcester Polytechnic Institute, and they created a machine learning tool based on image recognition models. So basically, this is, for those of you familiar with genetics, you can get a sequence, you go to BLAST from NCBI, submit the sequence, and it tells you what it matches to. So this is a BLAST, but for MEL curves. So you submit the CSV file, that is the output from the machine, from the qPCR, you hit submit, and it gives you what species you have in tube 1, in tube 2, in tube 3, with a confidence, a certain confidence. All of these ones are above 0.9, which is our threshold that we have now. And if it, for example, for this one, it gives you 0.48, 0.57, it's either a species that we don't have in the database, a variant that we don't have in the database, so you either rerun it or sequence it. But if you have a 99% match, you can be confident that you're looking at the right species and the right curve. So it's a very streamlined and easy process for anybody to use, because this is not really made for a scientist to use it. It's more for a police or a law enforcement officer anywhere in the world to use. So we have a website where you can have manuals, you can have a video tutorial, you can download the supply list to purchase the primers, the reagents, the whole thing, and you can have a direct link to identify those curves with that user interface that we created. We're also doing it for tunas, so now you can identify different tunas with this thing. We tried it in Long Island with NOAA a couple of months ago, and we found mislabeling of incoming tuna into the US everywhere we looked at. Yellowfin tuna, we looked, and no, it was big eye tuna, it was something else. So massive misidentification cases. And now with funding that we got and our partners, we're moving this towards turtles, mammals. Meredith is doing a whole bunch of trying to dissect the supply chain to know exactly where things are coming from, where they're transshipping, where they're going to the dark web to find where they're being sold. So it's outside my area of expertise, but she really is a good, a great addition to the project. And with that, I just want to say that there are other tools out there that are very useful. This is a portable sequencer machine, or mean iron. It's basically this size, and you can take it anywhere and do a sequencing of a DNA tissue anywhere, but it's a little expensive and it takes time. But if you combine this with our tool, then you can screen, it says yellowfin tuna. You screen your whole container for yellowfin tuna. The ones that are yellowfin tuna, you don't need to sequence. And whatever is a mismatch, that you can sequence in the field and basically close your case with our preliminary test and like a more forensic accepted sequence. And basically there's no fooling the DNA, so you can close your case right there while you're doing your inspection. We have those little red cubes in different places around the world. So if we were playing Risk, I think we were being a good in a good case to win the game. The idea is to bring this technology in as many places as we can in order to try to close down the gaps in those borders that allow transshipment of illegal wildlife to go unnoticed and undetected. So just going back at the end for our theory of change, we now understand a lot better the supply chain. We increase international trade regulations. We have developed and deployed illegal wildlife trade detection tools that would hopefully deter illegal trade and promote stronger enforcement and compliance. And that would hopefully lead to a better conservation and sustainability of species. And this is just to mention that it's just by focusing on the right questions and on the right problems. So with that, just thanks to you guys for having me here, our collaborators, our funders. And if you have any questions, we have a few minutes that we can use for that. Thank you.
So technically we are at time, but do we have one question that someone would like to ask? One question?
I'll ask a question. So Hong Kong, everything apparently goes to Hong Kong. Is that creative processing center?
Yeah, so the fins arrive there unprocessed and dried, and then they process them there.
And supplemental question to that is that when did it start in Hong Kong? Was that before 1987 or that when it was built in UK handed over to China, or was it before that?
Hong Kong and China have always been a consumption center, but when they started to receive that big volume of fins was just before the 60s. And I think 70s, 80s, 90s, it really peaked. And I think it peaked around 2010. And now we see a decline in volumes, but I think those volumes are a representation of a declining of supply, not just a decline in demand.
I think we better close it at that, because
All right,
we might have another class.
thank you.
Thank